Hello bachcho, let's read the question. The question says here consider the following statements. So there are two statements given here. You have to choose the correct option from these four options. So first one is given there hybridoma cells are shifted to a medium deficient in nutrient which cannot be synthesized by the myeloma cell. Then this medium allow the selection of the hybridoma cell. So let's see. So which one of the following is correct? So first of all, as we know, the antibodies derived from a single cell clone of the cell. So the antibodies they are derived from the single clone of the cell. Single clone. Single clone of the cell which recognize only one kind of antigen. So it recognize only one kind of antigen. So these are known as monoclonal antibodies. So these are known as monoclonal antibodies. And the major step for the production of the monoclonal antibodies with hybrid culture, these are mentioned below. That means first of all a mouse rat are sometimes. So यहाँ पे क्या होता है, a mouse rat or some other animal they are injected to a specific antigen. So any animal are inject a specific antigen against which the antibody are required. जो antibody required होता है, जो specific that antibody is required, a specific antigen is injected to some other animal or to the mouse or rat. Then that animal starts developing the antibodies. So that an animal have started developing some antibodies against the antigen in B lymphocyte cell or in the spleen. So the antibodies are created in the B lymphocyte and B lymphocyte cells in the spleen and the spleen of the animal. This is removed and its B lymphocyte of the cell, this is isolated. And similarly the cell producing bone marrow cancer cell, these are isolated. That is the bone marrow cancer cell, these are also isolated, and these cells should not able to. So that means this bone marrow cancer cells, these are the myeloma cells. So when the myeloma cell are attached to the hybridoma cell, this is known as the attached to the antibody producing cell, that is normal antibody producing cell, that is the lymphocyte. This is known as the hybridoma. So their fusion is known as a hybridoma. They are shifted to a medium or deficient nutrient which cannot be synthesized by the myeloma cell. So let's see. After this, these cells should not able to synthesize their own nutrients. So this is not synthesized their own nutrient and the two major type and the two types of cell, that is myeloma cell and antibody producing cell, these are known as the hybridoma, as I discussed in this part. And the entire culture is shifted, this entire culture is shifted to a medium deficient in the nutrient, and also they are shifted to shifted to a medium deficient in the nutrient. They are needed by the myeloma cell. That cannot survive. So first statement is correct. Then second statement is let's coming to second statement. After this in this medium all the unfused myeloma cell, they are die and only the hybridoma cell are survive. So only the hybridoma cells, these are survive. These are survive, and the surviving hybridoma cells, these are allowing to multiply. These are allowing to multiply separately, and each clone is tested for the ability of the production of the desired antibodies. So for that, this medium may allow the selection of the hybridoma, because the unfused myeloma, they are died and we select the hybridoma cell only. So this is also a correct statement. Then both the statement are correct, so option C is our correct answer. Both statement 1 and 2 are correct, and also this statement 2 is correctly explained about the first statement. I hope you understood it. Best of luck.